Hello, aspirants. My name is Shankar Banerjee. I'm the founder and educator at Battle Science Institute. And in today's video, we will be discussing about bacteriophage gene regulation. Now, before entering into the depth of it, let's consider how a bacteriophage it infects a bacterial system and how it chooses whether to perform the Lytic cycle or to enhance lysogeny. Let's see. Whenever it comes to, whenever, suppose a bacteriophage, it infects a bacterial system, as simple as that. It introduces its genetic material within it. I think all of you are aware of the mechanisms. I am not entering into the depth of that. So whenever the genetic element is introduced, either either the genetic element will utilize the host, either it will utilize the host machinery, will replicate itself and eventually cleave or lyse the entire bacterial system and invade another bacterial cell. Or else, what it can do, it can integrate its genetic element within the host, or it can introduce its genetic element into the host chromosome. These are the two possibilities. And how this is mediated? Let's see. Suppose when it comes to when it comes to Lytic cycle, when it comes to Lytic cycle, one, suppose a single bacteriophage, if single bacteriophage, it infects the bacteria. So whenever a single bacteriophage it infects the bacterial system, and suppose the system is supplemented with ample amount of nutrition, ample nutritional support, ample nutritional support, then only, in that scenario only, Lytic cycle can take place. So first, one single bacteriophage infecting the bacterial system. Second, system should be supplemented with proper nutritional support or nutritional backup. What is the reason? Because it can initiate rapid cell division. It can initiate rapid cell division. What is the purpose? So if these criteria are to be taken under consideration, this one criteria and this is the second criteria. Therefore, if these two criteria are fulfilled, then rapid cell division will take place and then only the cell can initiate the Lytic cycle. But when it comes to lysogeny, when it comes to lysogeny, first, not multiple bacterial, I mean multiple bacteriophage infecting a single bacterial cell, multiple bacteriophage infecting a single bacterial cell, and nutritional backup is poor. The nutritional backup is poor. Therefore, what will happen? Rate of cell division is slow. Rate of cell division is slow. Whenever these phenomenon occurs, whenever these kind of phenomenon occurs, then only there lies a possibility for a cell to enter lysogeny. Then only there lies a possibility for a bacteriophage to enter lysogeny for maximum rate of survival. For maximum rate of survival. So first of all, for a lytic cycle, single bacteriophage, one bacterial system, ample nutritional support, and rapid cell division. This is the criteria. For lysogeny, multiple bacteriophage, single bacterial cell, poor nutritional setup, rate of cell division is slower. If these are the cases, then that will lead to lysogeny. What is the purpose? Ultimate purpose is to survive. The ultimate purpose is to survive. All of you must note it down. Now, coming to the mechanism of it. Coming to my first slide. There lies promoter sites, operator sites all over the genetic material, within a lambda phage. But we will be focusing upon a few that is actually essential in order to initiate lytic or lysogeny. Here I have listed a few of it. Let's see. So it starts with C3. Then comes PL, that means promoter at left hand side. Then comes a protein called C1, a gene called C1, which is also known as lambda repressor, and which is associated with lysogeny, Next comes PRM, which is another weak promoter. PRM, which is another weak promoter. RM means repressor maintenance. Downstream of PRM lies PR. That means promoter on right hand side, because there is a series of genes on the left hand side. For left hand side it is PL, for right hand side it is PR. And we must remember its promoter sites. If there lies certain operator within these promoter sites, then if these operator sites are present on the R, that means right hand side end, then the same operator sites will be present on the left hand side as well. This should be kept in mind. Next, coming to proprotein. Proprotein means it is associated with light cycle. Proprotein is associated with lytic cycle. And then comes our C2. This C2 gene product, this is also associated with lysogeny. So, for lysogeny, till now, we have observed here lies C3, then C1, and then C2. All these three are associated with lysogeny. And for lytic, we are getting proprotein. And there lies another protein I haven't introduced over here, which is N protein. That is also associated with lytic cycle. So, now let's break it down. Whenever PRM, we are initially focusing on PRM. Let's see. It's PRM, which is a weak promoter. We have came across the term called PRM, which is repressor maintenance. And we must know that PRM is a weak promoter. Within PRM, but we have got we have majorly within PRM, we have got majorly operator region 3. Within PRM, we have got majorly operator region 3. This OR3, that means operator for right hand side. Jehetu right, that's why right hand side. And region 3 means there must be region 1 and region 2 as well. That is why it is stated as region 3. On the other hand, that means for PRM, that means promoter for repressor maintenance, we have got under PRM a site, an operator site, which is OR3. As simple as that. Whereas PR, which is promoter for right hand side. Within promoter of right hand side, we have got majorly, we have got majorly OR1. That means operator for right hand side 1. So for PRM, we have got majorly operator for OR3, and within PR, we have got operator side for operator at right hand side 1. Apart from these two, apart from these two major segments, there lies certain portions, extra, excess. There lies certain excess portions within both PRM and PR. Majorly OR3 for PRM and majorly OR1 for PR. Apart from that, we have certain excess regions as well. This excess overlapping region, this excess overlapping region is considered as operator for right hand side OR2. Operator for right hand side OR2. And this kind of phenomenon is similar that of EL as well. So that means what is happening over here? PRM is there. PRM has majorly OR3. PR is there. PR has majorly OR1. And apart from that, there are certain portions excess for both PRM and PR. This portion, this excess overlapping segment is called operator for right hand side 2. These segments are to be noticed because eventually in our next part of the discussion, these portions will appear very frequently. That is why I'm just providing an overview regarding what is associated with lytic, what is associated with lysogeny, what are the promoters, which are the strong promoter, which are weak promoters. Within promoter, what are the operator regions we have got? Because eventually the gene product will bind to these operator regions and they will promote either lytic or lysogeny based upon the environmental condition I have just discussed in the beginning of my class. So let's see. So let's say in terms of in terms of lytic cycle, in terms of lytic cycle, that means what are the criterias? Criteria number 1, single bacteriophage lambda. Second, medium supplemented with proper nutritional backup. And third, we have got Rapid cell division. So these three criteria suppose is being fulfilled. If these three criteria are fulfilled, then what will happen? Then synthesis, synthesis of protein, synthesis of protein will occur. What is the location of the protein? Let's see. So this is the location of the protein. The RNA pol will bind the promoter site and it will eventually synthesize the protein. As simple as that. We all know that this protein, we need to know a bit of its structural orientation. This protein is a homodimer. This protein is a homodimer where each subunit, each subunit bears three alpha helices. Each subunit bears three alpha helices. Helix 2 and 3. Helix 2 and 3, they majorly associate with the DNA element. Which DNA element? I mean, which portion it interacts with? We must keep a thorough investigation regarding the fact that this protein product must bind with the operator region 3, must bind with the operator region 3. Now, a technical fact is among these two helix 2 and 3, helix 3, helix 3 of it, it has helix 3, it has, it normally bounds with the major groove of the DNA. It normally bounds with the major groove of the DNA. And helix 2 stabilizes the protein DNA interaction. Protein DNA interaction. That means they form a helix loop helix interaction. They form a helix loop helix interaction. Now let's see. So first of all, we have our protein. This protein normally binds with the DNA element which is operator at right hand side 3, which is a part of TRM. And not only that, based upon its structural variability, it is a homodimer where each of the subunit it bears three alpha helices. Among these three alpha helices, helix 2 and helix 3 are mainly associated with DNA binding. More precisely, helix 3, it binds with the major groove of the DNA, and helix 2, it performs the protein DNA stability. It performs the protein DNA stability. So this is all about protein. Apart from protein, there is another protein which is N. There is another protein, I haven't mentioned it in my picture previously, N protein. All of the aspirants must comment below regarding what is the function of N protein in terms of Lytic cycle. All of you must comment What is the function of N protein in terms of Lytic cycle? Hmm. So we have got our CRO protein, and this CRO protein, what it does, it binds with the operator region over here. And this phenomenon takes place when there is a proper environment for initiating the Lytic cycle in terms of lambda phage. But the fact is, this binding of CRO, OR3, this binding of CRO with OR3, it hinders, it hinders the binding of another crucial protein, the binding of protein with OR3, it also hinders the binding of a crucial protein which is associated with lysogeny, that is C1, which is also called lambda repressor. Before entering into lysogeny, let's talk about how this C1 protein is generated. Before knowing C1, we have to know about another protein, which is C2, which is C2. Whenever medium is, whenever medium is supplemented with less nutritional backup, less nutritional backup, then what happens? And delayed cell division, delayed cell division, then what happens? DNA Pol, sorry, not DNA Pol, RNA Pol synthesizes C2. RNA Pol rather transcribes C2, which undergoes translation and form the C2 protein. The C2 protein, this C2 protein, interestingly, is a regulator. This C2 protein, interestingly, is a regulator. What it regulates and how it regulates? Let's come to the first picture. So we are talking about this C2. We are talking about this C2 protein. I haven't mentioned another promoter. This C2, after getting transcribed and translated, what it does, it on its downstream, it on its downstream binds with another promoter site, which is PRE. PRE. Once we have got PRM, and secondly we have got PRE. PRE, that means repressor establishment. Repressor establishment. This is also a weak promoter. This is also a weak promoter. So C2, after getting synthesized, it interacts with C2, after getting synthesized, it interacts with and binds with PRE. C2, after being synthesized, it interacts with PRE. And this kind of interaction of C2 with PRE, this kind of interaction of C2 with PRE will promote the cell to synthesize, will promote the lambda phage to synthesize C1. So C2, After getting synthesized, it binds with the PRE, which is a weak promoter, and this kind of binding will promote the synthesis of C1, which is stated as the lambda repressor. So this is the overall story of C1 getting synthesized. Now C1, after synthesis, it has to bind with a specific operator and has to hinder the process of Lytic cycle. That is the overall purpose. It's lambda repressor or C1, whatever may be. It has, based upon if we consider the structure of it, it is composed of 236 amino acids, and they appear in the form of a dumbbell where the linker is about 40 amino acid. Now each of these units, they initially form a dimer. Each of these units, they initially form a dimer. This dimer, this dimer has high affinity towards OR1. This dimer has high affinity towards OR1. Let's see where OR1 is. So C1, after getting synthesized, shows high affinity towards OR1. And how it binds? In the form of a dimer. After its binding with OR1, what will happen? More and more C1, will be synthesized. And if more and more C1 is synthesized, then it will also occupy the regions of OR2 as well. It will also occupy the regions of OR2 as well. So, if suppose in the beginning, initially, low C1 concentration, high affinity towards operator region 1. So therefore, if this is the operator region 1, then in the form of dimer, they are binding with it. But as the concentration is increasing, what is happening? Just side by OR1, we have OR2 region as well. Let's say this is our OR1 region, this is our OR2 region. So OR1 occupied by two dimers of C1. Nextly, concentration increases. OR2, though OR2 region doesn't have that much affinity for C1, but still, as C1 concentration is high, compared to normal, it will eventually bind with the OR2 region as well. And how this kind of binding will take place? By means of C-terminal domain. By means of C-terminal domain. That means this is our CTD and this is our NTD. So interaction will always take place based upon CTD. And these two will come close to each other, forming a structure like this. These two will come close to each other, forming a structure like this. This is how overall SAMR of C1 will function. This is how the overall SAMR of C1 will function on binding with both OR1 and OR. OR1 and OR. Now, to this point, the operator region 3 till now, up to this point, the operator region 3 is free. Because RNA pol has to bind with it in order to synthesize more and more C2. Let's say C1 is there. C1 is getting synthesized. Now C1 getting synthesized and it binds with the OR1 and OR2. And RNA pol can only move. RNA pol can only move up to this point. RNA pol can only move up to this point. Why? Because PR is maximally occupied by synthesized C1. So there is no space for protein to bind with OR3. Suppose this is our structure. Here lies our C1 protein. This is our PRM, which bears our region of OR3, and here lies our PR, which has portions like OR1, and this is overlapping region is our OR, and after that lies our protein. This is the overall structure. Now C1 is getting synthesized. When C1 is getting synthesized, it shows high affinity towards OR1. It will bind. Now the concentration of C1 is gradually increasing. So therefore it can also bind with the OR2. And their binding is occurring like this. They're interacting with each by based upon CTD. Now OR2 and OR1 is already blocked. But OR3 is free till now. But just keep a deep attention over here. Now OR3 is still free right now. But C1 synthesis takes place by means of RNA pol. C1 synthesis takes place by means of RNA pol. Now RNA pol, as it is synthesizing C1, it cannot exceed RNA pol. It cannot exceed operator region 1 and 2. Why? Because it is blocked by C1. In order to synthesize pro, it has to cover OR2 and OR1 region. But as C1 is blocking that region, pro cannot be synthesized. And if pro cannot be synthesized, then lytic cycle cannot proceed. As simple as that. So RNA-pol is strictly fixed. RNA-pol is strictly fixed at OR3, initiating lysogeny. Initiating lysogeny. I hope everyone is clear with this point. Then that when C1 is synthesized, why not Pro cannot be synthesized eventually? Because when C1 is getting synthesized by means of RNA-pol and eventually by means of translation, C1 will bind with, it shows affinity towards OR1. It will bind with it. Now RNA-pol will eventually synthesize more and more C1, and this increased C1 concentration will eventually occupy OR2 region as well. Now OR1 and OR2 are blocked by means of C1 protein. When C1 protein is blocking that region, then RNA-pol can only proceed up to OR3. But protein is located apart from protein. Sorry, downstream of PR. Protein is located downstream of PR. So RNA-pol cannot reach to that region by crossing this portion because it is blocked by C1. This is how lysogeny will result. This is how lysogeny will result. As simple as that. Now the fact is, suppose, suppose C1 concentration has increased up to 40 to 50% than it should be. Then what will happen? Suppose C1 concentration has raised more than 40 to 50%. So at this rate of concentration, what C1 will do? C1 will even occupy operator region 3. C1 will even occupy operator region 3. And at the same time, it will activate a protease. It will activate a protease within it. And that protease will initiate the auto cleaving of C1. আবার বলছি, C1 concentration is high. Normally, blocking C2, OR1 and OR2 is okay. But whenever the concentration rises more than 40 to 50% of the usual case, then what will happen? Then it will eventually occupy operator region 3 as well. And if OR3 is occupied, then what will happen? An auto cleavage mechanism will result within C1 itself, and that will cleave the entire protein concentration. That will cleave the entire protein concentration. This is how the overall C1 is regulating itself. This is how the C1 is regulating itself. Apart from, so one thing we have understood, C1, it is associated with auto regulation. Concentration low, lysogeny. Concentration moderate, lysogeny. binding, binding OR1, moderate, binding OR1 plus OR2, concentration high, more than 40 to 50% than usual, lytic. Why? Because autocleavage system will be activated. Now, apart from C2 or C1, there lies a protein, a gene called INT, which is integrase. This integrase will help. This is also associated with lysogeny. It will help the genetic element of lambda phage to introduce itself within the host chromosome. That means integrase will initiate the process of recombination, forming prophage. Integrase. So we have T1, we have what will happen based upon its concentration. Apart from autoregulation, apart from autoregulation, here lies another two mechanism. There lies two other mechanisms. Here lies an ATP dependent protease. ATP dependent protease called FPSH. This FPSH, what it does, it has the ability cleave. It has the ability to cleave the C1 protein. Hence, lytic cycle will result. Problem is, this FPSH, which is a ATP dependent protease, can also be inhibited. This FPSH can also be inhibited by what? By another lysogeny initiating protein, C3. C3 can hinder the activity of FTSH. C3 can hinder the activity of FTSH. So C1 can autoregulate itself based upon its concentration within the bacterial cell. As simple as that. But the problem is, apart from its autoregulation, there are other certain mechanisms involved. There is a protein which is FTSH, which is an ATP dependent protease, which has ability to cleave the C1 protein, and that can initiate lytic cycle, because cleaving of C1 will help the synthesis of crow-protein. Then crow-protein can bind to operator region 3, and hence lytic cycle will result. But the problem with FTSH is there lies another lysogen initiating protein, which is C3. C3 can block or hinder the activity of FTSH. C3 can block or hinder the activity of FTSH. So this is also a kind of regulation that is ongoing apart from the regular processes. So previously, I have asked you to comment about N protein. Now the second question, all of you must answer. What is the function of Q protein? What is the function of Q protein? All of you must comment. And how it is regulating process of lytic and lysogeny. So let's summarize the entire thing now. First, we have got a series of promoters as well as operators and specific genes. Based upon environmental condition, a cell can either enter lytic or lysogeny. As simple as that. Lytic cycle is associated with protein. Lysogeny is associated with C2, C1, C3, and many more. Now, whenever the overall nutritional support within the medium is high and the cellular division is rapid, then we have came to know that lambda phage will initiate lytic cycle. During lytic cycle, what is happening? Protein is getting synthesized. And this protein will bind with operator region 3. Where is it located? Within PRM. What is PRM? Weak promoter. Repressor maintenance. Within OR3, it binds. As simple as that and initiates the process of lytic cycle. But the problem is, whenever situation is not appropriate, that means when nutritional support is less and the rate of cell division is poor, by this time, lytic cycle will result. So the lambda phage will immediately switch to lytic cycle, from lytic to lysogeny. Now during lysogeny, another key component is C2. This C2, after getting synthesized, it will bind with another promoter region which is PRE, repressor establishment. This PRE is also a weak promoter. This kind of binding of regulatory protein C2 upon PRE will help to activate the synthesis of C1. Now C1, after getting synthesized, it shows high affinity towards OR1. OR1 is a part of PR. OR1 is a part of PR. Now, initially the concentration of C1 is less. So it easily binds with the OR1 and we all know the structure of it. But as the concentration increases, it will eventually occupy operator region 2 as well. It will eventually occupy operator region 2 as well. Just like this, operator region 2 as well. Now, as operator region 2 is also occupied, RNA pol cannot proceed further. RNA pol cannot proceed further. So it has to synthesize C1 only because OR2 and OR1 is already blocked by C1. So protein cannot be synthesized. Hence, lysogeny is the only option left. Now the fact is, whenever the concentration is maximized, suppose 40 to 50% more than it is supposed to be within a cell, then what will happen? Then it will initiate a auto-cleavage system. The C1 protein will initiate a auto-cleavage system and that will cleave the C1. As a result, what will happen? Then only lytic cycle can be resulted. Then only lytic cycle can be resulted. That's it. Apart from that, there is another mechanism which is there lies a ATP dependent protease called FTSH. This FTSH has the ability to cleave C1 protein. Whenever C1 protein is cleaved, we all know that protein can be synthesized, which can bind with operator region 3, which is a part of TRM, and it will eventually initiate lytic cycle. As simple as that. But there lies a problem, because the beginning of these gene sequences has C3, which is also a lysogen promoting protein. C3 has the ability to cleave, not cleave, rather inhibit the ATP dependent protease FTSH. So as a result, the C1 synthesis can take place by the activity of C3 upon hindering FTSH. This is also a kind of regulatory mechanism. And I have asked two separate questions. One, what is the function of N protein and how it is associated with lytic cycle? And second question is, what is the function of Q protein and how it is controlling the overall process of lytic and lysogeny? Apart from this, there is also a phenomenon which is UV driven. UV driven. We all know that ultraviolet rays, they has an ability to hinder, to cause damage within the DNA. And if this kind of damage occurs, then the first protein that is synthesized is REC-A. So in response to UV mediated damage, in response to UV mediated damage, the first protein that is synthesized is REC-A. This REC-A has also the ability to cleave C1. The REC-A has also the ability to cleave C1 that will also promote lytic cycle. That can also promote lytic cycle. So this is how overall process of lytic and lysogeny will result. So the key points is from where the question can appear. What are the environmental conditions for lytic and lysogeny first? When lytic is activated, how come the binding of protein with that of our OR3 takes place? Means the structural detailing of protein. Third, how C1 is regulated. This is third probability. Fourth, why excess concentration of C1 is hindering the process of lysogeny. And fifth, how come FTSH is mediating lytic and lysogeny? These are the 5 takeaways from this entire video, and which is a question prone area regarding CSIR-NET or GET or DBT-BET. Thank you for today. And if you find interesting as well as informative, then do share with all of your batchmates. And if possible, like, share, and subscribe, as I'm providing an immense effort into generating all these kinds of things, trying to reciprocate to you. And always remember, stay focused, build concepts, and track progress. Me, Shanko Banerjee, founder and educator at Vital Science Institute, signing off.